Hi, today we'll be talking about hereditary for this particular chapter. So over here, there'll be a few things that we'll be talking about. This will be split into a few series so that we could actually have more discussion about it. First of all, let's look at what is hereditary. So hereditary traits, right, is basically these characteristics that can be passed on from one generation to another. So what this actually meant is, let's say, from your parents' generation, let's say they have brown eyes. How were these these characteristics of brown eyes being passed down to, let's say, the children or the offsprings having the similar brown eyes is an example of hereditary traits. So some examples will also include like hair type. Is it curly? Is it straight? Shape of the earlobe, meaning is it attached or not attached? Eye color, we have different variations. We have blues, brown, green. Face shape will be the sharpness of it, the chin shapes as well. Ability to roll the tongue, this is actually more of a genetic kind of thing. Like some people can do, some people couldn't do at all. Skin color, and of course blood type. So how is this actually discovered? This was discovered by Mendel. He's the founding founder of modern genetics, and what he did was he studied monohybrid inheritance in peas. So why peas? Because peas, right, the life cycle to growth is actually very fast, and he can actually plant a lot of them depending. It's just limited by the number of space. So when we try to study genetics, right, we want to actually have that masses to in order to actually draw some form of um correlation, or at least some observation that this is the similar pattern that we are observing. So genetics is the study of the inheritance of the characteristics by the transmission of genetic material from one generation to another. So for Mendel, what he does is he would actually maybe take the pollen from some of the plants, from plant A, and then put it to another plant. So it allows cross pollination, and from there actually observe what are the characteristics being passed down to the offspring. Sometimes he might also have done the pollination to the within the same plant, so that's self pollination. And from there study what's the characteristics of their offspring. So what is monohybrid inheritance? So monohybrid comes from the word mono, and second word is hybrid. So mono means there is only one set of characteristics. Okay, however, this characteristic would have two contrasting form. Now what is meant by this? So let's say we use hair color. So hair color is one characteristic, because for hair you can have curly, you can have straight, you can see short, long, everything. So these are all the different characteristics of hair, but for here we are just focusing on color of the hair. Then when it comes to contrasting form, two contrasting form meanings these two forms are very different and they are distinct from each other. For example, black hair versus white hair. So these are what we call contrasting, and there's only two forms. It's not restricted to say like, okay, there's black hair, there's brown hair, there is green hair, golden hair, and white hair. No, so in this case, for our study for our bio for Sec 4 is mainly one characteristic having two contrasting form. So this characteristic, right, is each controlled by a single gene, and that each gene will consist of a pair of alleles. Now alleles later on you will see, right, is represented by letters. Okay, one letter. And each of the letter, one of it is a capital letter, let's say capital A, the other would be a small letter. So one's a capital letter, the other is a small letter. Okay, so what this actually meant is a capital letter will represent it to be actually a dominant allele. Dominant meaning something that is overpowering, something that is more superior. In this case, it's something that can be expressed. That means you can actually see it physically from the characteristic. Versus recessive. For recessive, basically these are generally weaker in expression. So for example, if let's say I have the dominant allele for hair color to be black and the recessive allele for hair color to be white, this would mean that for the hair to be black would mean that the allele for it will definitely have the allele for black in color because it is more dominantly expressed. Okay. So for what he did was he actually crossed between purebred tall peas and purebred dwarf peas. So in this case, right, we could see that for any characteristic, okay, in this case we are talking about a height, you will need to, it is usually made up of two alleles. Okay, so do take note, the characteristic of height is made up of two alleles. And if it's something you see, and let's say we use capital T to represent the allele, the dominant allele for tall, and a small t as the allele for dwarf, or in other words, shorter height. What would then be the characteristics representation of purebred tall peas? It will be capital T, capital T. Why capital T, capital T? Because it is purebred. If they never give you the word pure, then it can also be capital T, capital T, capital T, small t. Because it contains one of the allele to be stronger than the other, hence it is big T, small t. But we'll talk more about it later. For purebred dwarf plants, we are talking about small t, small t. So what happens is he planted the seeds from the cross and he observed that the F1 generation, so this is the F1 generation, is all tall. So when you talk about all tall, right, in this case, later on we will look at the crossing of the genetic diagram. It will actually be represented by one big T, one small t. Now, then later on what he does is he took the offspring, that means like an incest kind of thing, to cross with each other. So you cross it with the same offspring, which is all capital T, small t, which in the way you look at it tall, and you realize that the traits are different, in which there are the proportion of maybe three out of four plants, three of them are tall, one of them is short. So then he asked the question like, why is it having this kind of observation, shouldn't be all tall? Because for genetics is passed on from the parents. So now the thing is, when I take the offspring F1 generation and both our parents are tall, right, I should ideally see all my plants to be tall if this holds true. But why is there a difference for one of them? Why is there a dwarf? So then there's the hypothesis as this shortness of gene must have resulted from the initial parent. That means like the F1 generation's parent. So then the gene would have passed on from the tall in some way to the F1 generation, and then later on to the next one, which we call as the F2 generation. So what he did was he actually renamed the traits that always appear in the F1 hybrids, dominant and recessive. So in this case, right, what we can see is a few things. So first of all, from the parent's point of view, okay, the tall parent, and then there's a dwarf. Okay, you have a capital T and a capital T and a small t, because those are purebreds. Then later on, when you want to result in an F1 generation, this has to split into a capital T as a gamete, capital T as a small gamete, and so on. Now, why is this so? Because it is a big T cannot combine with a small t just like that. The number of diploid condition doesn't meet. So just like you, if you are asking for a mother that is very, very short versus a father that is very, very tall, their cells do not just fertilize suddenly. Instead, they must first produce the sperm and the egg by having the haploid condition. That's why they have to split. okay, to form the sex cells. So if I were to do it properly, these are what we call the parent's genotype. Then, which is still a cell, but then you want to form the sex cells, which is a gamete. So that's where you split to form the gamete. This is usually written in the form of a bubble and through the process of meiosis to form the sex cell. But of course, this one is plants lah. I mean, but then plants would also have to split out into its gametes, you see. So in this case, after that, you will get your F1 generation. So your F1 generation is where you are able to do the matching and the pairing, because this particular gamete must fuse with the other parent's gamete. So one thing to take note is you cannot allow the same gamete from the same parent to fuse each other. Like these two will not happen at all. Neither would this also happen, the fusion between this and this, because they are from the same parent. It's like the parents are having sex with themselves lah, which is not what we are looking at here. So there are a few ways to go about drawing this example. If let's say we do the combination of the lines one, right? My suggestion is you draw the line every cell, every of these one of the parents' gamete must have two lines. So if you draw it in this way, first, it's very clear already. Okay, one of the gamete cannot have cannot have more than two lines. So next, then you do the matching. Because this one line has to match with one of the gametes here, and this one has to match with the other gamete. So this will fulfill the F1 generation genotype. So if you look at how they actually match, it is a big T matched with a small t. A big T matched with a small t, a big T matched with a small t. Then comes the other one, where you have two lines again. One matches with the female one, and then it matches with the other one. So this will also still give you big T, small t, big T, small t. So this becomes the F1 generation genotype. And in this case, it becomes the phenotype. Phenotype is basically what you can see. This is tall, 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 tall. Why is it no dwarf? It's because the allele capital T is more dominant than the shorter dwarf gene, which is small t. So all the F1 generations are all tall. Okay, so this is the ratio. Now then after that we see Mendel, he took the F1 generation and he started to cross with each other. So the F1 generation parent is big T, small t, big T, small t. Both are also tall. So by right, his assumption, if he has never seen genetics before, all the offspring should be tall lah, because they're all physically tall. But what he realized is when he split it, so again the offspring undergoes meiosis to produce the sex gametes. So you got a big T, small t, big T, small t over here. These are the gametes. Then again the drawing comes in. Okay, you first draw each of the gamete to have two lines. So one rule of thumb is I usually end the lines underneath a particular gamete, so that the referencing, right, I know that it's always neater rather than everywhere it goes. So this is what I do. So the next one I will want to aim is somewhere here and here. And that will be my I will want to aim somewhere here and here. So that's where I will draw the second line. One here, one here. So each of these from one, so just have to choose one parent. Start with only one parent. Okay, you can do the right one or the left one, it doesn't matter, but start with just one parent, draw the two lines first. And make sure that they are positioned underneath each of the gamete. Then after that what you do, you match them up. Okay, you match them up with the opposite parent. So this is one. What you will get is the genotype for F2 generation. So capital T, capital T, capital T, small t. Then the other one, if you match it up, the other one is I tend to combine them. I tend to put them at the same point here so that at least they are also neater. So this one we'll see that is capital T, small t, capital T, small t, small t. Now it's a rule of thumb that you have to put the dominant allele first, then the small allele. You cannot put small t, big T. That's wrong. Okay. Then you go by the phenotype, which is the characteristic again. So because it has all those with the capital T one, they are all tall. Because it's the dominant allele. But because there's no capital T, it's small t. So that it's actually recessive, homozygous recessive. Hence, this is actually a dwarf. And therefore, the ratio is out of maybe 100, right? You will see that 75%, which is 3 of the plants being tall and 1 of it is being a dwarf. So this is generally how he observed through a genetic diagram. Okay. If you don't really feel comfortable doing this drawing, then the other one you can use is a Punnett square. It's like a tic-tac-toe. But then what you do is you just put one of the parents allele up and then the other parents allele down. So this is the F1 generation. Then you do a cross. So the cross is something like that. Down. Down. So this is capital T, small t, capital T, small t. Then the other one, so this becomes capital T, small t, capital T, small t. And what you can see is all of them are having the same as this one. Okay, if you are looking at the other one, then the ratio will be something like that. Again, draw the tic-tac-toe. You have your big T, small t from the parent, big T, small t from the F1 generation parent, like we saw here. Okay. Only one box will have one from the outside. So this one will only have one. Then you do the matching. So you have big T, big T, big T, small t, big T, small t, small t, small t. How the naming will look like is this way. Big T, big T, big T, small t, big T. Then the other side, big T, small t, small t, small t. Then you will also get the same ratio as the one below. So these are some of the techniques to go about drawing lah. So Mendel's model of hereditary is where the hereditary factors are responsible for the transmission of characteristic and that each of these characteristics is controlled by a pair of factors in the cell. If two factors are different, only the dominant factor will show its effect, meaning the one with the capital letter allele as the allele. Okay, this one varies by question to question. So the two factors separates with segregate during gamete formation, and each gamete will only contain one factor, which is the law of segregation. The random fusion of gametes will help to ensure that the zygote will contain two factors. So for Mendel's law of segregation, this is how it actually looks like how I just draw for you. This is an example of a Punnett square. Okay, and that is the general basis of hereditary. Now let's look at some of the basic knowledge for studying it. So this portion is more towards of how do we actually do the naming convention for hereditary. This one do rewatch a few times because you need to be very familiar in order to move forward. So a chromosome is this rod-like structure that is visible in the nucleus during cell division and is mainly made up of DNA. So if you remember in chapter 2, which is cell, we realize that inside an animal cell there is a nucleus. So this is the nucleus. And inside it you have multiple chromosomes. Okay, there's quite a lot. In humans there's about 46 of it. And for it to be visible, right, it has to have been tightly condensed, which we actually talk about it in the chapter of cell division. So when it's condensed, it will be something like that. Okay, like a cross like this. And this is one chromosome. So there's altogether 23 pairs of chromosomes or 46 chromosomes in a normal human cell. And all of these are made up of DNA, which is deoxyribose nucleic acid. We will talk more about this in the next chapter on molecular genetics, because DNA comes from a few words. So D comes from deoxy, meaning the ribose sugar. The ribose sugar that is made up of the whole molecule doesn't contain oxygen, the oxygen atom. And then nucleic acid is the other one. So there's a ribose sugar, there's a nucleic acid, and the lack of oxygen. So in a typical chromosome, this whole thing is one chromosome. Okay. There's a gene. So gene is a unit of inheritance bonds on a particular locus position of a chromosome. Meaning in a particular chromosome, you can have multiple genes. But these genes can be coding for some things like, let's say, like hair color, can be hair type, it can even be your skin color, etc. So in a typical chromosome, you can carry many different types of genes. Okay, so that's what they meant by a unit of inheritance, where we are talking about only one gene. And when we talk about locus means the position of the gene. Okay, and this is actually a small segment of DNA in the chromosome that will control a particular characteristic of the person. So if let's say the chromosome is talking about hair color, within the gene, maybe all of the allele that is dominant inside is black hair, versus another chromosome that it has white hair. So that's some of the examples lah. So alleles are the different form of the same gene. That means both of it are coding for, let's say, height. So it's the same characteristic. However, they are represented in alternative forms, meaning this one maybe is talking about tall, the gene for being tall, and the other one is about dwarf, okay, being short. Okay, and they occupy relatively the same position on the pair of homologous chromosomes. The meaning of homologous chromosome means the chromosomes that pair with each other. Okay, and usually why we say this is because one of the chromosome would come from the mother, one will come from the father. So there's the maternal, and the other one will come from the paternal. That's why they are in pairs. Okay, and we usually use letters to represent the alleles. So for homologous chromosome, right, the meaning of homologous chromosome meaning is that the chromosomes are in pairs, and that one chromosome in the pair comes from the male parent being the paternal, okay, and the other is the female, which is the maternal. So one way to remember it easily is that M represents for mother, P represents for like papa, the father. Okay? And they have exactly the same sequence of the gene loci, but not necessarily the same allele. So they are only coding the same characteristic. Like let's say these two gene loci are talking about hair color, but one can be black hair, the other can be white hair. But they have to talk about the same characteristic, which is hair color. Okay. So together, all of these would actually represent the how your chromosomes will actually be packed in the nucleus, and within the chromosome you can multiple layers of genes, and each gene will have different alleles being expressed. If it's the same allele type that's being expressed, then you will definitely have that kind of variation, that kind of view and expression already. So the phenotype of an organism refers to the observable traits, and the phenotype of an organism is usually influenced by two things: the genotype and the environment. So genotype is mainly the genes. The environment is mainly like external factors like the sun. If let's say it is a very you put yourself under the hot sun every day, your skin will be very toned and dark. Okay. And so the genotype is actually the genetic makeup of an organism that is inherited from the parents. So this is something that you cannot see. This is found only inside the gene. So just now you saw the word homologous chromosome. Now the thing is, it's no longer homologous, it's called homozygous. So the meaning of homo, right, means the same. So when we talk about homologous chromosome, we are talking about how the different chromosomes are in a way similar in a way. But when we talk about homozygous, we are not talking about the chromosome, we are talking about the alleles. Okay, so meaning alleles, let's say over here, capital T, that means they are the same. So they are homozygous dominant. Whereas if let's say they are all small t, they are both recessive. So you say that they are homozygous recessive because they are the same. So this word refers to the allele and not the chromosome. Then comes in the meaning of heterozygous. Heterozygous means different. The meaning of like let's say you are heterosexual means that you like someone from the opposite sex. Okay, so the meaning of hetero means different. Then if let's say someone says you are homo means you prefer someone of the same gender. Okay. So for a trait, if the two alleles are controlling the traits, they tend to be different. So like only in Sec 4 syllabus, there is only two alternative forms that you will be matching. So therefore, there's only three types of combination that you'll see. You only see this combination, where it is homozygous dominant. The second one that you will see is homozygous recessive. And the final one that you will see is heterozygous, which is different. Now the letters can change, but just take note that when what they are meaning, right, is that homozygous dominant means both are capital letter. Then homozygous recessive means both are small letters. Heterozygous means that you have one capital and one small letter. That's all. Okay, so the alleles can exist in dominant or recessive form, where the dominant allele is represented with an uppercase, whereas a recessive one is actually represented by a lowercase. So this was already being discussed just now, where a dominant allele will be able to express itself in both homozygous and heterozygous condition because they are they are the one that is stronger in expression. So regardless if they are different, like let's say if capital T, of course you'll be tall. But heterozygous capital T small t, then it will be tall also because of the capital T. Okay. Next, recessive allele will only express itself in the homozygous recessive because there is no dominant alleles at all. Okay, so when we look at this, right, what we can see is for the phenotype, right? Phenotype is something physical that you can see. For flower Q and R, even though R is heterozygous and that Q is homozygous dominant, it will still be reflected as pink because they have the dominant allele of A. However, for P, because it is a homozygous recessive, in terms of the gene, therefore it will remain white. So in this case, I can assume that the dominant allele A is to represent purple flower, whereas the recessive allele is actually white flower. So A is purple color. Small a, which is recessive, you call it white color. Okay. And that genetic models can be used to explain how alleles are passed on to the offsprings to predict. So this one is generally the variations that can actually happen. So it's a lot of drawing and things like this. So for this one, my suggestion is go and memorize how to draw this, especially these labels, because in exams this can come out as a structure question. It's usually worth 2 to 4 marks. Okay, you will not be you may not be given helping words or what, but you have to draw from scratch. So first of all, usually you will see the format to be more or less similar. It starts with phenotype, genotype, gamete. Then the generation will be again phenotype, genotype, and then phenotype. So refer to just now how I actually explained the terminologies. So first of all, you start with the genotype of the parents. Then of course, for the parents to actually mate or what's not, the only thing that can cross is the gametes, which is your sex cells. So meaning your sperm, your ovum, that contains half the number of chromosomes. So it is split according to the letters. So you have 1T, so someone can draw. So usually, as a rule of thumb, right, a circle represents gamete. Do not go and draw a circle for your different genotypes because you'll be marked wrong. Okay, so the circle represents gametes, and then from there, you will actually form the genotype. Now, this is not a correct representation. My suggestion is to actually draw something like this, even though it will all give you the same combination. In exam, this is the one accepted. Okay, you draw something like this one you won't get the marks at all. Then, of course, when you cross again, do the same combination, you get the answer. But do you see that why is this so messy? Because they didn't follow the rule of thumb that I shared lah. So the thing is, actually they sort of did lah, but it can be quite messy because they went to draw the arrow and what's not. And so one of the suggestions, like I shared with you all, just make sure that you stick with one parent first to draw the lines. Then after that you combine, it'll be easier to match. Whether you want to leave it in a ratio method or percentage is also fine. So how do you do it in a percentage form if they ever ask you as a follow-up question? So there's a total of 4 units here, and 3 of it is tall. So just take 3 tall over the total number of combination, which is 4, times 100%. This will give you 75%. So 75% will be tall, then the remaining 25% is dwarf. Do take note that total for this must always hit 100%. So the Punnett square is something that I also did share with you all just now. But there's one thing that I really like is how they actually label the characteristics at the bottom. So this will also give you another way to actually view it. So how to go about doing it if I were to split it up in terms of the combination, this is how the whole Punnett square looks like in terms of the information that is being given. So if I were to color it, the red one is here, the pink one, the orange one, and finally the green one. Okay, so this is the kind of combination that you need. Of course, more practice will make more sense. And then from here, you can take a look. The ratio is definitely just 3 tall is to 1 dwarf. Okay. So I will pause here and then we will be discussing the next few slides later on. But do revise your work first.